Drug discovery is broken is one of the things that everybody in this room continuously hears, and I continuously hear it. And being inquisitive, I want to know, well, why is it broken? Who thinks it's broken? What are we going to do about it? So just to satisfy my curiosity, show of hands, how many people in the room agree with this statement? Okay, not many. And how many disagree with the statement? Far more. Now that's really, really interesting. Really interesting. So why have we got ourselves to a point where some people think that drug discovery is broken? Well, this actually came out in 2012, so it's a bit out of date now. But as you can see, and I expect many of you familiar with this, what is the return on investment for drug discovery? And drug discovery is becoming increasingly inefficient. So you can see here, it's costing too much money and it's taking too long to get drugs to market. It's been a bit of an uptick here, and we haven't got any data or at least I haven't got any data for the last 6 years. So there is signs of optimism, but clearly there is a problem here. I think we shouldn't be in denial about it. Whether it's broken or not, I personally don't think so, as I will hopefully convince you in the rest of the talk. But it is a challenge. So where's all this time and money and effort going? Well, here we have the 3 stages. So we have over here the biology, which is all done mainly in universities, institutes, government-funded, charity-funded, getting figures for how much that actually costs and how long it takes. Impossible. So that's why there aren't any here. And then within the preclinical space, which is where I think there's the most opportunity for cost savings and time savings, we have perhaps the most challenging problem here. So we've got 5.5 years that on average these things are taking in preclinical. And only 35% are getting through, so we've got a high attrition in preclinical. And it's costing enormous amounts of money, $800 million plus just to get something through preclinical. And that's to the point where it can just go into the clinic. And then when it goes into the clinic, again, we're faced with similar problems. So 8 years in the clinic. We're down only 10% coming through, as we all know, and it's the figure that is sort of bandied around by everybody in the industry, that only 10% of the drugs going into clinical get out the other end and get onto the market. It's a real problem. And of course, the cost again is absolutely enormous. So you put all that together, and you're looking at average 13.5 years. From getting your lead compound to something on the market, only 3.5% of those get through. And then the cost is just absolutely astronomical. The rewards are great, but the costs are huge. So the thing that we agonize about, and people in this room, I'm sure it's the audience that oh, sorry, before I go on to the agony bit, this, for me, is really interesting. This is a piece of research which is done by looking at how many publications that were funded by the National Institutes of Health are coming out per year. And you can see the enormous increase here. So we're nearly up to 100,000 publications per year through NIH-funded programs, which is absolutely astonishing. And I'm one of those people who probably like yourselves in the audience, I continually evangelize about the discovery in biology and the new things that we can do, and the single-cell sequencing and the iPS cells and the transcriptomics, genomics, proteomics. All of these discoveries are hugely exciting, and we all get a bit emotional and teary-eyed about it all. But the problem is this doesn't seem to be translating into good drug discovery, efficient drug discovery. And it's a real paradox. And in any sort of paradox, we like to find someone to blame. And I'm sure we all here do the same thing. We're always trying to get, well, who's to blame for all of this? You know, what is going wrong? And we agonize about this continuously. So we say, well, is it regulation? It's just too difficult an environment to get drugs onto the market. Is it MHRA? Is it FDA? Now things are improving. We all heard about orphan status drugs, compassionate use, fast track. All these things are great. So I think the environment there is actually improving, but it's something that we hear again and again and again. Lack of biomarkers. So the holy grail for, if you go back 10, 15 years, everything was about biomarkers. It was about personalized medicine. It was about segmentation, stratification of patient groups. This was going to be the answer. And that has kind of succeeded, but not to anywhere near to the degree that we had set the expectations for. And then there's, of course, the blame now that's going around on wrong target selection. Classic there is sort of amyloid tau, and I hear figures, I don't know if they're true or not, rumors that the industry spent well over $30 billion completely wasted cumulatively on chasing these targets, which turned out to be the wrong targets. In hindsight, lack of druggable space, again, are we just trying to drug the same kinase pocket again and again and again? Where is the new druggable space, and where's that going to come from? And have we found all the druggable space yet? And I hear that. Biological complexity, it's just too complex. There's too many off-target effects. I hear this again and again and again. This is why we've got the high attrition rate. All the low-hanging fruit's been picked. Again, people say blockbusters have all gone. Much more difficult now to find drugs that really work in a blockbuster type fashion. And these are the blockbuster really is the answer to that. The lack of blockbusters was the biomarkers and the personalized medicine. That hasn't actually come to fruition. Now, I have my own little theory about what's happening here, and I blame these little critters here. And these are meant to be, we thought, little fairy mammals, but of course, Douglas Adams proved to us back in 1979 they are actually hyper-intelligent, pan-galactic super beings. And a lot of our sort of research and assumptions, and don't get me wrong, I fall for this just as much as everybody else, and I'll show you that in the slides later. But a lot of our assumptions are based on mouse models. And the number of times I've heard, well, we've put it into mice, and it cures cancer in mice, in this mouse model. Well, you can inject bleach into some mouse models, and it cures cancer. So these little creatures here, fairies they might be having a laugh at our expense. So that all sounds a bit depressing. But it depends who's it depressing for. I think the most thing it's depressing for is all of us as patients. All of us will be patients at one point in our lives. And the lack of good drugs coming through, effective drugs, especially in diseases of aging, your cancers, your dementias, your cardiovasculars, this is where there's a real need. and we just don't have the drugs coming through. Saying that, of course, I'm an optimist, and also I see opportunities, and there are opportunities for entrepreneurs, scientists, investors. So I'm going to spend the rest of the talk just thinking about where those opportunities might be for those people that are interested in that. Well, biology, as we've seen, is fabulous. It's booming. With all the new tools that are coming out, I'm quite envious of anybody who's doing research now and trying to do a PhD, because it's quite easy to get publications. The tools are fabulous, the proteomics, the genomics, the single-cell sequencing. There's so much opportunity there. The thing is, though, it's a bottomless pit of money if you're trying to do drug discovery. So that's best, and it definitely is best left to charities and government. And I've tried to do my own bit here. So I did my postdoc from 95 to 98, working on breast cancer research at what is now the Gurdon Institute, which was back then the Wellcome CRC Institute, with Professor Tony Koutsaridis. And I was having a beer with Tony a couple of years ago, and he said, Jonathan, it's really frustrating. We've got this fabulous biology and in the university, and all these drug companies have got these fabulous drug libraries and chemistry, and the two just are not talking efficiently together. And I said, you're pushing on an open door here, Tony. I totally agree with you. And that's how we had the idea for the Milner Institute. So that's specifically set up to foster collaboration between early stage biological discovery and drug companies, biotech, pharma. So that's in Cambridge. So this is the area that I really want to talk about, and it's the preclinical area, where there are, for me, huge opportunities. So what could be those opportunities? I've listed a few of them here. Just as a way of background, I started my new career founding life science companies and investing in life science companies, probably back in 2007 with the first investment in Horizon Discovery, whilst I was still the CEO of Abcam. I stepped down as the CEO of Abcam in 2014, and since then I've been doing it as a full-time career, just spotting opportunities in this preclinical area, and then investing and helping those companies to thrive and prosper. So one area for me which I find really interesting is in artificial intelligence, machine learning, whatever you want to call it, and everyone's got a different definition of the two, so they're kind of used interchangeably there, especially in repurposing and rare disease, which I'll talk about one of the companies involved there. Breakthroughs in finding new druggable space, trying to crack this problem of where are the new drugs going to come from. Human cellular disease models, and we're having a revolution now in iPS cells that can be differentiated into cellular models, and these are being increasingly used, and the market for those is exponentially increasing. And this will hopefully help the problem of not having to go into mice and get misleading results. Immunotherapy, massive, savior of the industry. I've got colleagues who work at Addenbrooke's Hospital, and they've said to me, Jonathan, this is the first time we've actually had therapies that are curing people, albeit it's at a low rate. We're at the very early stages of it, 10 and 20% cure rates we're seeing in some lung cancers, for instance, which are before completely intractable. So this is a hugely exciting area, and we've only just scratched the surface there. And then looking at drug targets, if we've got the wrong drug targets, where are the drug targets? Where are we going to find them from? And I want to talk a little bit about that as well. So the company that I want to present, so I've got these sort of 5 areas, and they're just a snapshot of what I'm involved with. My portfolio is about 20 companies. I sit on the board of 10, And I didn't have time today to present all of those. So I've just done a selection of a few of those that I can just briefly introduce you to. And if you want to take them further, by all means, I'll give you the contact later. So there's a company called Helix, which I'll introduce. A company called Foremost, which I started with Chris Torrance and Ashok Venkataranam there. Alpis I'll introduce, Lift Biosciences, and Shift Biosciences. And then, of course, there's the clinical. And this is an area which, for obvious reasons, the expense, the expertise, etc., is best left to big pharma to do. And the one thing that I hear again and again and again from companies, especially startup companies, there's a little bit of naivety in the preclinical area, is, I've approached AstraZeneca or whoever it might be, and they don't seem to be really that interested, and it's really frustrating, or they're leading me along and all that. Well, of course they're not. This is the area here where we should be presenting really good, solid drug packages to pharma at this stage, so that there's far less risk for them to go into the clinic. They're not stupid, and they don't want to be spending their time, money, effort down here when it's much better spent on really good data packages that are ready to go straight into the clinic. And this is what I tell my companies again and again and again. It's all about getting a really good data package. And if you go to pharma with a good data package, it's worth a huge amount of money, and they are always going to be interested in that, always. So let's shine the spotlight on Helix for a moment. I'll introduce you those people here. So Tim, he's from Belgium. We won't hold that against him. He is a neuroscientist, and then he did an MBA at the Judge Institute, which is where I met him a few years ago. And he always had a passion for treating rare disease. And he's the founder of the Cambridge Rare Disease Network as well. And he came to me with this idea of a company absolutely focused on rare disease and repurposing using artificial intelligence. And then it got even better because the chairman is Dr. David Brown. And David Brown's a legend in repurposing, having led the program on repurposing Viagra. So he's sort of a god of repurposing, if you like. And I'm sure many of you in the audience know Dave very well. So he joined as chairman, and I joined as the first investor director. And behind Helix is this thing we call HealNet. And HealNet is this massive repository of information, of public information and private information, of transcriptomics data, proteomics data, drug data, rare disease data. 7,000 rare diseases in there. There's nearly 15,000 drugs in there. There's all the publications in there. And you put all this together in a way that I have no idea how it works, but there are people that do, thankfully, and we know it works because we've tried it, and it works again and again and again. And apparently, this is what it looks like. So there we go. That's what it looks like. And part of it, which I can just about understand, is what we call transcriptomics matching, but it's only part of it. Where you don't, the beauty of this is you don't actually need any pathway information or biological information. You simply take the transcriptomics data from your cells which are grown in the presence of the drug, of which there's quite a lot out there, with the cells which are disease-type cells, and then you look for imaging, so you can correct the transcriptomics profile of the disease, matching it with a drug. So it's done completely blind of any biology. That's just part of it. But then the power of it is to take the brains as well of people, such as Dave Brown, who can then actually pick out the really interesting things that this HealNet is spitting out. And why are we so excited about this? Because we can do it cheaper, faster, and it's more successful. And we know that repurposing, there's very good evidence out there that repurposing is so much easier for obvious reasons. So we can bring the cost right down to a fraction of the discovery cost, traditional discovery cost. And in fact, that 50 million, I think, is high. These are figures that are out there. At Helix, we want to do that for under a million to get the cost right down. The time, our record to date has been about 18 months to go for the preclinical phase to get something from a lead ready to go into the clinic, which is our Fragile X program. And of course, the success rate you'd expect to be really high, considering that these are all drugs that are already gone through phase 1, at least, safety procedures. So 25%, I think, is pessimistic. I think we can actually do better than that. We're only at the start of this journey. As I said, we've got 10 programs in this current phase. We've just raised $10 million with Balderton Capital has come on with as one of our investment partners. And we're going to do 10 programs. Then we're going to raise a whole lot more money and try and get this up to 100 programs. So it's ambitious, but because of the model and because it's repurposing, because it's using this very novel technology of HealNet, we can do it. Looking at new druggable space, this is a company that I got involved with purely because of Chris Torrance. So Chris Torrance is, for me, a genius, and I like working with really clever people. And he was the one who, back in 2007, said, "Jonathan, let's go for a drink in the pub in the Eagle in Cambridge." And he said, "I've got this piece of gene engineering technology out of the University of Washington." I said, "Oh, tell me about it." Anyway, cut a long story short, out of that came Horizon Discovery, which is now listed on the stock market, AIM stock market. It's done very well. And Chris left that because he felt that he wanted to do something more in the drug discovery space rather than the tool space. And he met up with Ashok Venkataranam, who works at the LMB at the University of Cambridge. And Ashok told him about his drug discovery platform. And out of that was born Foremost. So the three of us got together and we started Foremost. And it all relies on having really good feel for what is druggable space. And at the beginning of it, we've got here libraries of different shapes. And in fact, the first library that came along was Ashok's archibacterial library, because he had a hunch that they would be three-dimensional shapes that are found nowhere else in archaea that could have really strong interactions with human proteins. So that's the library. And then it goes through a phase of screening, and we use in vitro phenotypic screens, which is these two parts here. And that enables us to look for cryptic sites that close and open. And one of the first programs was on RAS, which is traditionally sort of undruggable. And we found a cryptic site there, and we've got a compound there that's in lead optimization. That contrasts with the traditional way of doing it, where you've got X-ray crystallography. So that's very, very exciting, and we've got a number of programs there. But perhaps even better well, not even better, but I suppose as unique, if you like, as the platform, is the way that we're going about this. And we've called it, because one day, jokingly, I was saying to Chris, this is really interesting. What do you want to do, Chris, in terms of the business model? Drug discovery is very risky. Single assets are dreadful. We don't want to invest in those, all those reasons we've just discussed. He said, I've got exactly the right idea. He said, I'm calling it the goose golden egg model. And that's where you've got a platform technology that can identify leads. You polish the egg to get it sufficiently of an attractive nature that it's going to be picked up by venture capitalists, and then you spin those out. And we've just done the first of those spin-outs, which is a mismatch repair gene called Neo4. And we've got a number of other 4-type companies in the pipeline here, as you can see. And these are all our partners here. So this enables the platform technology company to be independent and then to get royalty and license fees from these spin-out companies in this goose golden egg type of model. So, next I want to introduce company Alpis, and we have here in the audience Dr. Mark Cotter. And Mark came to me a number of years ago telling me about this intriguing technology for differentiating cells using 2 safe harbor sites where you can put the transcription factors in one site and the operating factors in the other site in 2 safe harbor sites, for which Mark did the research in his lab. So Mark's a clinical lecturer in the neuroscience department at the University of Cambridge, and again, a genius. And for me, the funniest thing is that Mark likes to relax by doing surgery, real surgery on real patients, and that's his form of relaxation. So it's quite an honor to be working with him. So what's the idea here? Well, at the moment, of course, Yamanaka, we know about that and how it's revolutionized our thinking on differentiation. And this is a traditional way of doing it here. The way that Mark and Alpis have discovered of doing it is something that we call forward programming. And you can take an iPS cell and you can go straight to a fully differentiated cell within a matter of days and not having to coax it through weeks and even months in the traditional route. I was just blown away by the power of this technology, and I really like it because it means that you can theoretically have any sort of cell type here, as long as you know what the transcription factors are that you're putting in the cassette. You can just put those in, and away you go. You've got your differentiated cell within a number of days. And we've tried it, and it works, and we're very excited about it. And just so that you believe me, we've got a video here of skeletal cells, which have been transfected with the transcriptional cassette for skeletal cells. And here they are differentiating. You can see them here. And they're differentiating to the point here where they're actually going to contract. And they're contracting to such a degree that they're actually lifting themselves off the plate. So it's a really powerful demonstration of what this technology can do. So enormously exciting. Next, in the immunotherapy space, and sorry, I want to mention that if anybody wants to pick up with Mark after he's here. In the immunotherapy space, I bumped into a chap called Alex Blythe. Alex is a serial entrepreneur, and his mother, unfortunately, died of pancreatic cancer, and he wanted to do something about it, and he started reading about it. and came across Professor Q. And Professor Q had been working with innate immunotherapy and had shown that in a mouse model that he could cure cancer, in a mouse model at least, using these innate immunotherapy with a technique which is called leukocyte infusion therapy, hence LIFT, with a very special class of neutrophils. So this has opened up the way for allergenic treatment, and we're going to have we're just about to go into clinical trials for pancreatic cancer, and we're going to have the world's first neutrophil cell therapy. So enormously exciting again. And then I want to spend a little while talking about this company here, which the more that I get to work with this chap here, Dr. Daniel Ives, the more intrigued and blown away by some of the science that is coming out here. So again, genius, came out of University of Cambridge with a PhD in neuroscience. And he'd been working on mitochondria. So he was at the mitochondrial unit at the University of Cambridge. And he was working on something called mitochondrial heteroplasmy, which, I'll be honest with you, I have never heard of before I met Daniel. And he explained to me what it was, and I got more and more intrigued by this mitochondrial heteroplasmy. I started to do some reading around it, and I started to think, and Daniel was sort of instrumental in this, in changing my attitude to thinking of mitochondria as perhaps having there's a rich source of drug targets in there for a whole variety of diseases. So what Daniel was working on, and this is, by the way, this is all published and out there, and there's nothing controversial here, is that as we age, our mitochondria accumulate damaging mutations, and if you look at aging cells, they have a higher proportion of aged mitochondria in, and a lower proportion of healthy mitochondria. So Daniel was working with a class of glucose mimetics, which when you added them to the cells in vitro at least, you got this intriguing result, which was that you could shift, hence the name shift bioscience, the proportion of healthy mitochondria and decrease the proportion of unhealthy mitochondria. And by unhealthy mitochondria, these chaps over here, these have been implicated in all types of diseases, principally actually diseases of dementia, Parkinson's, Alzheimer's especially. And in fact, it was Parkinson's that Daniel was first working on. And you can show repeatedly that in Parkinson's patients, they have a high proportion of damaged mitochondria with large amounts of mutations in very specific parts of the mitochondrial genome. So he'd applied this and then taken that observation out of the university and with a class of glucose mimetics that we're now starting to use in a preclinical setting, looking for new drug targets. It looks as though some of these are operating at the level of hexokinase 1 and hexokinase 2. And I mentioned this is where these little creatures they seduce us, and I'm only showing you this for a bit of fun because we put some of these glucose mimetics into the treated group. So at the moment we've only raised a very small amount of money, and mice are quite expensive. So we put some drugs into some mice on the right here. So these are the shift drugs. These have been treated for 9.5. These are 9.5 months old, rather. I think they've been treated for 2 months. And these are the controls here. And the intriguing thing is that it seems to be rescuing the aging phenotype in that hair growth, as you can see here, is much more than the hair growth around the inoculation site here of the controls. And as I say, it's intriguing, and these little creatures, they beguile us, and they sort of suck us in. But we have some evidence that this could be working in a way that is also in anti-aging. And there's a lot of research and huge amounts of money, interest in anti-aging, as you're all aware of. So whether these drugs are acting on that pathway remains to be seen, but we've got a number of programs going on now with progeria cells, etc., in that area. And the exciting thing is that I mentioned discovery, golden age of biology. Steve Horvath published back in 2013 the epigenetic clock of aging, which applies to all tissues. And there's about 350, I think 390 now discovered methylation sites, which gradually change over the lifetime, meaning that you can very precisely say how old somebody is simply based on this assay that Steve has invented. So we've now got the opportunity to be able to use that in a context where we can put our drugs into a progeria cell model and look at and see if they affect aging. And why is aging so important? Well, the depressing thing is, having hit the age of 53, then my chances of disease just massively increase. So from the age of sort of 50 onwards, we are all prone to, and much more likely to get these diseases of aging. And I think the industry, in a way, has missed a trick here, because and also regulatory bodies have missed a trick. So they don't classify aging as a disease, but the biggest risk factor for any of the diseases of aging, your cancers, your dementias, your cardiovasculars, it's all, it's the biggest risk factor is obviously aging. So if you can actually find things that are slowing aging, you can stop these diseases. Quite obvious in retrospect, but something I think we've missed a little bit of. And that doesn't necessarily mean huge increases in longevity, which is the criticism, I think, that all of this aging research has is, well, why do you want people living forever? It's just a burden on the health service. World's population is too great anyway. It's a ridiculous idea. Well, no, it isn't, because if people can be just living another sort of 10, 20, 30 years healthily, and then have a couple of years maybe of morbidity and disease, and then drop dead, or preferably be on the tennis court at 110 and then drop dead, wouldn't this be a wonderful, wonderful thing? I'm evangelizing here, and I'm getting a bit ahead of myself just because of these mice. So they're doing it to us again, but hey, you've got to hope. Okay, well, in summary, we do need to make drug discovery faster, cheaper, and less attrition. It's obvious, and this is one of the reasons that this conference is so important. This is why we're here. I'm really optimistic for the future. There's these new life science preclinical companies that are addressing the problem. They're using all the new discoveries in biology that are coming out of labs like Mark's here. And of course, it's going to present for people in the audience who are classifying themselves as scientists or investors or entrepreneurs. There's huge opportunities here for you guys. So I'm incredibly optimistic about the future, and I'd like to leave it there. Thank you.